Hello everyone, I'm Shivangi Patel, a member of Shivi Forensics. Today we will discuss some MCQs on DNA. So, when we hear the word DNA, it may sound like a very easy topic because everyone is familiar with it. But even in DNA, there are some important basic points that we must remember. So, today we will talk about those key concepts through simple MCQs. So, the first question is, who proposed the double helix structure of the DNA molecule? Watson and Crick proposed the double helix model of DNA in 1953. Their structure showed two twisted strands and was based on Rosalind Franklin's X-ray diffraction data. This model explained how genetic information is stored and copied. So the answer is Watson and Crick. Now moving on to the second question. Which of the following nitrogenous bases is not found in DNA? Uracil is not found in DNA. It is present only in RNA, where it replaces thymine. DNA contains adenine, thymine, guanine, and cytosine. So the answer is uracil. Now third question. In a DNA double helix, adenine forms how many hydrogen bonds with thymine? In the DNA double helix, adenine pairs with thymine through two hydrogen bonds. This A double bond T pairing helps maintain the stability and proper base pairing of the DNA structure. So the answer is two hydrogen bonds. Now moving on to the fourth question. The process of DNA replication is considered to be. In this question it is asking that the DNA replication process, what to be considered. So DNA replication is semi-conservative, meaning each new DNA molecule has one parental strand and one newly made strand. This was confirmed by the Messelson-Stell experiment. So the answer is semi-conservative. Now the fifth question is, which enzyme is responsible for unwinding the DNA double helix ahead of the replication fork during replication? Helicase enzymes unwind the DNA double helix by breaking hydrogen bonds between bases, allowing the replication fork to open during DNA replication. So the answer is helicase enzyme. Now the sixth question is, Okazaki fragments are formed on which strand during the DNA replication? Okazaki fragments form on the lagging strand because it is made in the small pieces, not continuously. The DNA is copied in short segments away from the replication fork. So the answer is lagging strand. Now the seventh question is, the fragments of DNA on the lagging strand are joined together by which enzyme? So the DNA ligase joins the Okazaki fragments on the lagging strand by sealing the gap and making the DNA strands continuous. So the answer is DNA ligase enzymes.